Hello students, so today we go for the next concept of electron transfer, that is iron sulfur protein. Iron sulfur protein. So this is also one type of electron transfer protein. In the last video we have seen that cytochromes, cytochromes, how they transfer the electrons in the mitochondria. So similarly, so today the next protein. So this protein also we see in the living cells as well as the plants. Okay, so this iron sulfur protein, FES is also called as FES. So there have been two types here. So one is redoxin, redoxin, and one more is ferredoxin. Redoxin and ferredoxin. Okay, so these proteins are non-heme proteins. These are non-heme proteins. Okay, they don't contain porphyrin ring here and iron. So that's why it is not called. It is called as non-heme. Like in cytochromes, we had the porphyrin ring. So here there is no porphyrin ring, but the sulfur of cysteine is present here. Sulfur atom is present here. Sulfur is present here. Which is labile sulfur, that is called inorganic sulfur. Inorganic sulfur, okay. So which is found in the bridge form. So that we call it as labile sulfur. And one is cysteine, sulfur of cysteine. So this is going to make iron sulfur protein, is it right? And they are also helpful for the electron transfer or redox system. So wherever there is a redox type of reaction, like in nitrogenous enzymes, okay. So we have seen the reduction and oxidation there. In such type of mechanism, redox mechanism, we can see that such type of electrons, so one electron, two electron transfer, reduction, oxidation, wherever the process takes place in the living system. So you can see that this iron sulfur protein is also very much important. Their importance is very well here. So one by one today we will learn. That is, the first one is redoxin. So what is this redoxin? So this is nothing but this is a protein, iron sulfur protein, having the formula Fe1S0. Okay, iron sulfur protein having the formula Fe1S0. S0 means what? It does not have inorganic sulfide. It does not have inorganic sulfide. There is no labile sulfide, no bridge here, and that's why this is called as 0. So that is it has the sulfur, but it does not have the labile sulfur of the inorganic sulfur. That's why we call it as Fe1. So what is the structure? It's a very low molecular weight. Okay. And if you see the structure of this, it is a regular tetrahedral structure. It is having a regular tetrahedral structure. In this. Okay. So this is sulfur here. Okay. Then here is sulfur of cysteine, element of sulfur, and here sulfur of cysteine. Why? Yes. Sulfur of cysteine. Like this. And they are having the chain in this way, like this. They are going to be bound in this. Like this. So this is your redoxin. So low molecular weight, which is also very rare studied protein here. And this is also one electron transfer or acceptor protein, which is having a red color. So it's having a very red color, okay, because of LNCT, ligand to metal charge transfer. This protein is red in color in its oxidized form. So in oxidized as well as reduced form, the iron is present here in the +2 state. Okay, so +2 state, and it is red in color because of the LNCT, ligand to metal charge transfer, a regular tetrahedral, a unique protein here, very less studied protein, I can say, redoxin. So this is what the structure as well as the very few points on this structure, iron sulfur structure, and that's why it's not having any sulfur of inorganic labile sulfur, but all are sulfurs of cysteine here, like this. Okay, so this is what about the redoxin. Next we will go for ferredoxin. Hydroxyl is classified into different types here. Okay, so that's what we will learn in detail, because this is the one iron sulfur protein which is utilized in most of the process in mitochondrial process. Okay, mitochondrial electron transfer process. So they are having some different, that is, they are different types like 2s, 2fe, 2s, 4fe, 4s, or 3fe, 4s, like this. Or it is also written as Fe2, S2, Fe4, S4, Fe3, S4 protein, like this. Okay. And one more protein is there, that is called risky protein, that we will see in the later. That is called risky protein. Iron sulfur protein is called risky protein. Okay. So one by one we will see now. The first we will go for 2Fe, 2S. So how it is being made here? Okay. So first we will go for one by one. So Fe2 and S2, or also called as 2Fe and 2 sulfur. So name itself says here that it is having the 2 iron here. So like this, so 2 iron here, and 2 sulfur is bound here by D non-sulfur. So like this. So this is 1 sulfur, like this. And this is a back bond here, like this. Bond to the front of the plate here. This is 1 sulfur. And another sulfur of 16. Okay. This iron is bond with sulfur of 16, like this. Sulfur of 16, sulfur of 16, like this. This is the structure. Like this, and they are bound like this. So this is what about the Fe2S2. Okay, this is also used for what? If you see here, so it is EPR inactive. So this is the EPR that is having both iron here is having the antiferromagnetic nature here. Okay, they are anti-ferromagnetic nature. Okay, so +2 antiferromagnetic. So both cancels here. So 1, 2, 3, 4. So here it is 1, 2, 3, 4, 5. So both gets here cancelled, and they are EPR inactive. They are EPR inactive. Okay, and if you see the structure here, so they are also somewhat a tetrahedral structure here. So if you see here, so this iron both are in the regular tetrahedral structure, and they are helping for what? They are helping for 1 electron transfer. So it is in the form of what now here? Iron. Okay, 3 sulfur, iron, 3 here, like this. So they will take 1 electron. 1 electron, and one of the iron will get reduced here in the form of what? Iron, 3, sulfur. Okay, sulfur here, S2. You can write on the S2 also here, like this. So here iron 2, like this. So this is how both the iron is helping for the 1 electron transfer mechanism, and this is how one of the iron here is going to be reduced here when it is having the electrons, 1 electron transfer or acceptor or the giver here. So this is how the Fe2S2 sulfur, okay, 2Fe2S sulfur plays the role here in the living systems. Okay. Next we will go for the next, that is Fe4S4 sulfur here. Okay. So next we will go for the next sulfur, iron sulfur protein. So that is your 4Fe4S. 4Fe4S. This is also a typical type of iron sulfur protein here. So which is having the cubic structure. So this is having the cubic structure here. So I will draw here small structure here in this way, like this. So here corner opposite the corner position will be situated, will be placed by 1 atom here. So iron. So here it is sulfur here. So okay. So you can take it here. Sulfur, iron, like this. Okay. And this iron is been attached with the sulfur of 16. Okay. Sulfur of 16, like this. Okay. Sulfur of 16, like this. Here also sulfur of 16, like this. Sulfur of 16, like this. They are joined. This protein is joined. So this is the protein, that's called 4Fe4 sulfur protein here. For here, the oxidation state, if you see of iron here, so it is this 3, and here also oxidation state is 3, and now here it is in 2 oxidation state, and it is in 2 oxidation state. And that's why what will happen here, the electrons, since like in previous iron proteins, how they are arranged in the what is say Fe3 in antiferromagnetic nature. So here also they are arranged in the antiferromagnetic nature, where the electron gets cancelled, where the electron get cancelled like this. Okay, and that's why this protein is also EPR inactive. So they are not going to show you electron paramagnetic resonance. EPR inactive. So they are also EPR inactive. They are not going to show the resonance, paramagnetic or ESR resonance. In the resonance is not observed here in this protein. Okay, so how they are going to transfer this protein? So you can see here. So I will remove this part. Now how they are going to transfer the protein here? So we will take one example here. So here P4, okay, sulfur of 16 plus 4, okay, plus 3 plus electron again here plus P4 sulfur of 16 plus 4, okay, plus 2 electron plus P4 sulfur of 16, okay, plus 4, and this is plus like this. Okay, so this is a reduced form. This is the reduced form. Okay, and what is this? This is the oxidized form. This is the superoxidized form. This is the oxidized form. So what is here? So how is the situation here? 4 Fe. Out of this 3 Fe will be plus 3 state, and 1 Fe will be plus 2 state. Okay, so that's why it is called as oxidized form, superoxidized form. Okay, this is called as a superoxidized form because here 3 Fe is in plus 3 state and 1 Fe is in plus 2 state here. Okay, and next if you see here, so plus 2 state, so it has taken 1 electron now here. So from plus 3 to plus 2. So here what is happening here? So 2 Fe is in plus 3 state and 2 Fe is in plus 2 state. Is in plus 2 state. Absolutely in equal form. 2 Fe plus 3, 2 atom Fe in plus 3 and 2 iron in plus 2 state here. So this is how the situation is. So this is also one type of oxidized form here. But compared to this, it's for superoxidized. This I can say just oxidized form here. Okay. Now it will take another electron here, and you are going to get this here. Almost here, what will happen now? There is 3 Fe is in plus 2 state. Okay, and 1 Fe is in plus 3. 1 Fe is in plus 3 state here. So see, so 3 Fe, the majority which is the reduced form is there. Majority 3 atoms of iron is in the reduced form. That's why this is called a super reduced form. We call it as a super reduced form. We call it as a super reduced form. We call it. See, so like this protein is going to Fe4S4 protein is going to transfer the electron itself getting oxidized, itself getting reduced. Okay, so a type of redox reaction is going to happen in the mitochondrial cells of the living organisms, plants. Okay, photosynthesis we can see. Okay, nitrogen fixation process we can see this how they work. That's what the next protein what we are going to see. So that is a very beautifully is used in the nitrogen fixation. What is that? So next protein, so we go for the next protein, that is your 3Fe4S. Is it right? The next protein, that is your 3Fe4S protein. 3Fe4S protein. So where you can see here 3Fe1, this is 1 and this is 1 Fe. Okay. Like this it is been attached to the sulfur. So where this this protein is attached to sulfur of again 16 here. So here also sulfur of 16. Here also sulfur of 16. Okay, like this. Okay, and in between you can see another sulfur is being added here like this. Like this. So how many? 1, 2, 3, 4. So 4 sulfur and 3 iron here. 1, 2, 3 iron is there. Is it right? So in this one specialty is what now they are going to decline the protein like this, and this is this protein is used where in nitrogenase, nitrogen fixing. that this is used in the nitrogen fixation, this protein. Okay, so they reduce and we get the carbohydrates. So this is how in the nitrogen fixation when we see that, so we will see that this protein how it works in the nitrogen fixation. Is it right? So this is what now. So here one thing is that so three iron are there, in which 2 iron is in +3 state, okay, and 1 is in +2 state. 1 is in +2 state. That's why it is the EPR. So it is going to show you EPR active. So this is the EPR. So it is going to show you EPR active. Other 2 proteins was inactive, so they are not going to show the resonance structure here, electron resonance. But here you can see the electron resonance here, because the iron of one iron is not in a different spin state here. So +3, and 1 is +2 here. So same thing, this also helps in one electron transfer here. So when it gets reduced form, so what will happen now? So 2 will be in +2 state and 1 will be in +3 state. When it gets oxidized, so 2 will be in oxidized form and 1 will be in the reduced form. So like this, so this is how this protein is used in nitrogen fixing nature, and this is how it also helps for the one electron transfer system as a releasing as well as the acceptor protein here electron, and this is how they are structured here, in this form. Okay, and I said in the start of that one more protein is there, that is the risky protein. So what is that risky protein? We'll see that also. This also they will ask in the competitive examinations. Okay, risky protein, that is risky protein. Okay, what is this risky protein? It is similar to Fe2S2. Fe2S2. Okay, so like this. Sulphur, and iron. So this is what Fe2S2 protein we have seen. This is how it has been the first protein of the pyridoxine. So we have seen like this. But what is this rescue protein? So rescue protein is nothing but, so this is bound with the sulfur of cysteine. This is bound with the sulfur of cysteine. Yes. Now this iron is not bound with the sulfur of cysteine. This iron is bound with the nitrogen of histidine. So this is what here, nitrogen of histidine, and this also nitrogen of histidine, like this. And they made a protein. Like this. So instead of sulfur here, so this iron is bound with the nitrogen of histidine, and this protein is called as a risky protein. So 2 marks question will be there. Small difference, a small change here. So in Fe2S2 protein, we have seen that this iron is being attached to all sulfur of cysteine here and 2 labile. So this is what sulfur of inorganic sulfur or labile sulfur bridging. Okay, this is called as inorganic sulfides. They are labile. Okay, so that is what this is how we have seen the structure here. But in risky protein, what will happen here? These 2 nitrogens, this iron, instead of sulfur of cysteine, so they are going to be bound with the what? They are going to bond with the nitrogen of the histidine. So this is how the iron sulfur protein plays an important role here. Is it right? So like this, in this class, in this video, we learned regarding the electron transfer mechanism with respect to the iron sulfur protein. Okay, which is a non-heme protein. So there is no heme here like in cytochromes. Is it right? It's a non-heme protein where it has been classified into 2 types. That is your ubiredoxine, which is having the tetrahedral structure, low molecular weight, very rarely studied case here, which is also helping in what one electron transfer here. And the next is what? Ferridoxine. So ferridoxine is classified into different types here, like Fe2S2, Fe4S4, or 3Fe3S4, like this. Is it right? And one more protein, that is what? Risky protein. So all are very good electron transporters as well as electron acceptors. Okay, and they play a very important role in the redox type of reactions wherever it is required in the living systems. Okay, in our body system also we can see, in plant system also we can see. So plants are going to produce the fruits from the carbohydrates. How it is possible? Okay, so to produce the fruits, carbohydrates is required to break down the carbon dioxide in the presence of sunlight and water. So what will happen during this? So there is required of what? The reduction of the carbon dioxide, which is going to form the carbohydrate C6H12O6. Sucrose C12H2211. So this is what happening here. So that requires, who is going to provide the electrons for them? So this is what happens. Is it right? Nitrogen is, yes, definitely. So it will fix the nitrogen, is it right? So when fixing the nitrogen in the form of different salts, so that requires electron oxidation or reduction. That who is going to help that? So there is no chemistry behind that person going and doing the experiment and putting some reducing agent. Is it? Have you seen in any plants and you are putting some reducing agent into the plant so that we get the fruits? It's not like that, is it right? So inside it's body creations. Okay. So inside only we can see that in our body also we are not eating reducing oxidizing agents here to get the food oxidized with energy and all. So it's all who is doing it. It has been done by some of the proteins. Is it right? And this is what the proteins very beautifully we learned in the last video. That is cytochromes A, B, C, even in F. They mentioned in some of the process. So very nicely we have learned how they are, which structure they are having and how they work in the previous video. And today's we have learned regarding the iron sulfur Phes sulfur protein and its different types of redoxin and ferredoxins and different conditions. Hope you understood this concept of today's class. In the next class we'll go for the some other systems, other concepts of the bioinorganic aspects. Thank you. Have a nice day.